natural ecosystems and our ecocolumn have many substantive differences that can overall differentiate our data that we collected versus what we might have collected if we were to take data from outside. So one of the substantive differences that we noted was the sunlight. In a controlled environment, obviously, like the amount of sunlight is very strictly 12 hours of sunlight and 12 hours of darkness. But outside in the natural ecosystem, the weather is kind of unpredictable and has many different variations depending on the season and the weather for that day. So, an example would be like precipitation, such as rain or snow, or a day that maybe the sun is completely out versus a day that there's a lot of overcast and it's really cloudy, so that the plants aren't seeing direct sunlight. And this can have effects on the plants, as in our eco column where the plants were getting direct light for 12 hours, they were able to photosynthesize to the best of their ability because they were always getting that sunlight that they need to go through that process. And this overall affected our data because of the growth of the plants that we were able to see. Our tallest plant, I think, grew up to 62 centimeters. And I think that was our buckwheat. Yeah, it was our buckwheat. It was able to grow like 62 centimeters because there were no temperature limiting factors, as opposed to what we might have seen if it was in a natural ecosystem.
Yeah, and I feel like our buckwheat could have grown a little taller, or like any of our other ones could grow, or any of our other plants could have grown taller too, because at the beginning ours like grew pretty fast, I would say, and the light that we had for the like our light source just wasn't tall enough, so like our plants were like hitting the lights, and then like I guess the leaves would like burn and like dry
yeah,
up.
like
so.
once they like touched the light.
Yeah, and one of our squash flowers that actually grew pretty big dried up and fell off.
Yeah,
But
because it was so close to the light.
Yeah.
But this most likely probably wouldn't happen in a natural environment, at least not during a winter season, such as in a deciduous forest where we live, because the sun isn't so close and it's not always in immediate direct sunlight.
Yeah, and like leaves do dry out, but like
Yeah.
ours was like touching the light.
Yeah.
Yeah, and also like a lot of plants outside are seasonal, but that didn't really matter because we were in a room temperature area. Another substantive difference between the natural ecosystems and our ecocolumn is the nutrient cycling that we were able to monitor. So in the ecocolumn, we found our nutrient levels, so our like nitrate, nitrite, and ammonia would fluctuate dramatically between the different days that we recorded the data.
Only the nitrate and the nitrite would. The ammonia.
Just the nitrate and the nitrite.
In the aquatic chamber.
Specifically in the aquatic chamber, yes. And this is probably due to the simplified biodiversity that we can see in our aquatic chambers, such as the one allodia and the one fish that we have. Another characteristic that might have affected our data because of a substantive difference in the ecocolumn versus the natural ecosystems is the compact habitat. So because we were growing in the size of just the plastic water bottle versus an open space outdoors, the plants were confined to a small amount of area to grow. So obviously they were able to grow, and we saw that with the growth of our plants, but there was a lot of competition, and most likely more competition than we might have seen if we were to do this experiment outside, because outdoors there would be more room for the plants to grow. And in the Ecocolumn, they were kind of competing for the space, and they had to grow through. I think some of the plants even grew through like the filter on the edge of the bottle cap.
Yeah, into the filtration chamber.
Yeah, and it was kind of like going through the mesh going into the filtration.
Yeah, and I think it was our spinach that like where we planted the seed, it was like not where we planted it. It was like a little like
Yeah, like to the
more to the
roots,
side,
like-
and we started going up.
Yeah. So another thing that we can see in our terrestrial chambers, specifically in the soil in the compact habitat, is that the worms were likely highly influencing the nutrient levels in the soil, because it was in such a confined space that like whatever the worm was doing to decompose would affect most likely like the whole thing, as well as the pill bugs and the other the sow bugs and the other, what was it, like the potato and
the potato and the
Eggshell.
eggshells.
and dead leaves that we
Leaves,
put into.
And all the dead plants that got burned up by the light.
Yeah, and that mostly had a higher impact than it would outside, because outside, obviously those components would be able to spread apart, and because of the wind, they might be like picked up and brought to another area that wouldn't have any effect on the plants. But again, because of the temperature and the weather and the compact area that the plants had to grow in, they were highly affected by everything that was in the chamber.
So for the graph, the ammonia and nitrate across the age of the aquatic chamber, we can see that the ammonia was relatively low, and it was 0 most of the time, except for a little bump at like around like
The 20
like
day
20
mark.
day mark.
Yeah.
And that like represents a pretty like healthy ecosystem, because it's like 0. And then, but we do see a spike in nitrate at like, I'd say like 25, like 30 days.
Yeah.
And that can indicate that denitrification is not happening, because it usually happens in low oxygenated conditions and waters, which is true, because if you look at our water DO across the age of the aquatic chamber, we can see that it does decrease around that time too. So it like makes sense that denitrification might not be happening. But it can also indicate that there's increased nitrification, because as I said before, right before the giant spike, we had a little bump in ammonia, like increased ammonia, and since there's more ammonia available, nitrification can happen, or increased nitrification can happen, and that can increase the nitrate in the water. And like I said before, the DO had decreased from when we started collecting the data, and that could possibly be because our Alodia was not able to photosynthesize as well, because it was blocked by the rest of the chamber, likely because the light was like directly above and not like to the side or anything. So like the soil and all the other plants were just blocking the Alodia, the sunlight for the Alodia to grow, or to photosynthesize.
So our terrestrial pH versus our aquatic pHs differed by a lot, because our terrestrial pH was fluctuating every couple days, while our aquatic pH was mainly steady across the whole time. And while we're not exactly sure why this occurred, we theorized that since both of them the average is above the normal 7 pH, it's because the photosynthesis occurring in both chambers was taking up the CO2 and the carbonic acid that was released by like the decomposing matter and raising the pH. And then for the terrestrial chamber, it might have been the decomposing matter releasing that carbonic acid back into the soil that's dropping it drastically, and then the photosynthesis raising it back up. And then also the like night cycles, or in this case our simulated 12 hours of sunlight, 12 hours of darkness, was because photosynthesis was occurring, raising the pH during the day, and then respiration was occurring, dropping the pH during the night.
So in our plant growth, in our terrestrial chamber graph, we can see that plants for the most part were growing steadily over the days that we were taking data. And then towards the end of the growth period, they started to plateau and they started to stay at the same height for the rest of the time. And this is most likely due to the fact that, like I said before, that they had very limited space to kind of grow their roots. As we saw when they would start going into the filtration chamber. And then when the plants grew too tall and were touching the light or were getting too close to the light, so they were able to touch the light and therefore burn.
Yeah, and since we didn't want them to burn, we like moved it to the side a little so it wasn't getting as much sunlight or as much like direct sunlight as before.
Which also might have slowed down the growth than what we saw before.
Yeah, and the plants that started off low kind of stayed low because the canopy that was created from the other plants, especially the buckwheat and the squash, whose leaves got like really big, so they were kind of like shadowing the other plants, so they weren't getting as much sunlight.
Yeah, I think especially our cucumber didn't grow very big, and this kind of like was true for the whole time. I think because it took longer than the rest of the plants to grow, and like you said, they were covered, so they were a little less competitive than, say, the buckwheat plant.
We decided that our ecocolumn was stable as an environment because the multiple species diversity within the terrestrial chamber allows for a higher tolerance, and the decomposing matter that occurs within the terrestrial chamber just gets cycled throughout as fertilizer for the pre-existing plants that are still living. And the photosynthesis and cell respiration that occurs within the terrestrial and the aquatic chamber allows for the CO2 and O2 levels to be regulated within the environment. And the plants in the terrestrial chamber allow for energy to cycle with them and the allodia acting as primary producers for our ecosystem. And this keeps the balance of the ecosystem from becoming unstable, and due to it being a closed system without any outside factors affecting it, we are able to control everything that occurs. And it is sustainable as an ecosystem, but not for long, because we use the water from the aquatic chamber to water the plants in the terrestrial chamber, and then it gets filtered down back into the aquatic, so everything gets reused over and over. But one thing that isn't sustainable is the root space. As the plants grow, the roots take up more space, and then they get more cramped and aren't allowed to continue growing, as was shown with our graph. And the disruption within it could be the fish producing excess ammonia through pee and raising our ammonia to an unsafe level.
So how the terrestrial chamber affects the filtration chamber is that when we water it through the roots, some of the water gets soaked up by the roots, but the excess water, along with like the nutrients and the minerals, are washed through into the filtration chamber. And there's like a little mesh thing that we had at the opening of it, so not all of the minerals and stuff went through, but the ones that could go through would be filtered out in the filtration chamber. So the water gets purified, and then it will also filter through to the aquatic chamber for our fish. So as I was saying, from the terrestrial to the aquatic chamber, through the filtration chamber, the water gets filtered out, and then so it's cleaner water, and the water quality is better for it, and there's more oxygen able to get through to the water since the plants go through photosynthesis and cellular respiration, and so that helps with the living organisms in the aquatic chamber, which was our alodia and our fish, and that they can get the oxygen they need to survive. How the filtration chamber affects the terrestrial chamber is that with the filtered water from the terrestrial chamber, the filtration chamber filters out the water to be purified and clean for our fish, Sebastian. For our fish, Sebastian, but also we take the water from our aquatic chamber back into the terrestrial chamber to water our plants so that they can grow, and then the cycle just keeps going from the terrestrial chamber, and all the nutrients and the water that isn't absorbed into the roots goes into the filtration chamber again, and then it just keeps recycling. So how the filtration chamber affects the aquatic chamber is that the water that gets filtered out from the terrestrial chamber gets purified and cleaned and obviously filtered out, hence the filtration chamber, back into the aquatic chamber so that our fish can have oxygenated water to survive, and also the alodia too. And then from the aquatic back to the terrestrial, we take water from the aquatic, like I said before, to the terrestrial so that our plants can grow. And like John said before, this is a closed system, so none of the water that we use to water is from an outside source. It's from our Ecocolumn.
But it's really hard to differentiate between the chambers because it is just a cycle that keeps repeating, and since they're all interconnected, each one kind of overlaps with the other. So what affects the terrestrial will also affect the aquatic and the filtration chamber.
Yeah.